So welcome everyone to this symposium. It's from the neuroscience track of the Mind Foundation, and it's about addressing the challenges and the opportunities both that we have in bridging the gap between basic science and clinical research in psychedelic neuroscience. We have 5 speakers. Each one will give a talk first, and at the end we have a 30-minute panel discussion where I'll be happy to take all other audience questions as well as try to bring these speakers together. We have today first Professor Dietmar Schmitz, then Professor Robert Malenka, Dr. Torben, Professor Kim Kuypers, and Professor Olson. Professor Olson will join us online. He is in US and he couldn't be here physically, so he will give an online talk. And yeah, it's very early in the West Coast, but that's why we have him at the end, and he will also join us for the panel discussion. So I'm looking forward. And first I would like to invite Dietmar Schmitz, who is also my supervisor at the Charité. So Dietmar, the floor is yours.
Yeah, thanks, Prateep, not only for the start, the introduction, but also for the initiative of triggering this event, for the coordination, and for the chance, the opportunity to discuss with you some of our ideas and also some of our data. I decided to have a kind of a jump start. Could you show perhaps these slides? I prepared a few. I was trying to. It's a tile slide, which I would like to skip. Can I use then the remote control here? Is this something? Sure. This is another one. This is Torben. My name is Dietmar Schmitz. I'm the first speaker. It's not connected. Then I perhaps have a kind of a spontaneous few sentences. So I had a long-standing interest in, not directly into psychedelics initially, but into serotonin, which is a transmitter, or some people call it neuromodulator, and how this affects the brain. I actually started working on this, and now I realize I'm a bit older. This was more than 30 years ago, 31 exactly. This is also not me. Dietmar Schmitz, but I mean, this would be actually interesting. I mean, I could try to give your talk and then we switch a bit. Might be a bit more creative. This looks like the one. And we are working in different brain areas, and I will come to the kind of cellular effects and then the network effects of serotonin, which is a kind of the intrinsic ligand for all these, or many of these psychedelics. Here we go. Who am I? Here. And I will start with an anecdote. It's a kind of a personal encounter into this area. So I came from the west coast of the US, from Berkeley, San Francisco here to Berlin in 2002, roughly. And the president of the Humboldt University invited me to have a chat, and he's a physicist, a great physicist, and he said, well, Dietmar, we should really trigger interest in young people and students for our stuff we are doing. Natural sciences, all people do law school, and it's also important, of course, but it would be great to have natural science people, or medicine, psychology. And he asked me to coordinate with him and others a kind of a university for, this might be too close here, for kids, children. And this is a kind of a general thing I like to do. The perspective of kids on certain things. So I had a huge meeting, so 700 kids in the audience, parents, was in the kind of a cinema, plus teachers. And it was about the brain, and you know, this was primary school, so you had really young kids, 5 years old. They asked more about where's the brain located. And then older kids, 12, 13, they Googled me and they found out that I had just a paper published on cannabinoids, on cannabis, how this would affect the brain, endocannabinoids, and they asked me then how cannabis would affect the brain. So kind of broad range. I had first to synchronize all these kids, very noisy, and then I did a massive neurosurgery here. All of our kids here, and there was indeed a brain located, actually a YouTube thing, but it was fun. But what was so rewarding then, so I mean, this sounds perhaps a bit full of ourselves and arrogant, but we had lots of invitations afterwards, and we really introduced something also into schools. Yeah, it's not only me, but also other people going into schools. And one thing I received from a teacher from also here from Germany, from Münster, was an essay, 4th class, young girl. And it's in German, but I translated it in English. And yeah, it's perhaps my personal thing. I can laugh over and over again. Perhaps this triggers also your serotonin release. Let's guide me through this. With the brain, we think that we think. I mean, this is philosophical and also not so easy to prove. Yeah, we think that we think. We also need it for headaches. It's located in the head, right behind the nose. When we sneeze, it drips. And now the two last lines is best. The brain is a very sensitive organ. That's why most people use it very rarely. So I think it's great, and perhaps I should stop here, so hopefully the questions are then a bit easier. So now I also try to do science. And this is some people, I mean, there are all these things for all these molecules, the God molecules, the mood molecule, it's called sometimes the magic. I call it 5-hydroxytryptamine. That's a chemical form. It sounds a bit boring, but that's serotonin. I started working on it, as I said, in the beginning of the 90s. So there it was clear where it's roughly located. I have also a slide on this. So here's roughly the structures of those. And you see that these, I mean, it's a bit academic, the terminology, what is psychedelic, what's not psychedelic drugs, classical, non-classical. But you see the structure is, for some of them, the DMT, the psilocybin, and also the LSD, based on this structure here, which is an intrinsic ligand. And I thought I'd give you roughly a short introduction into serotonin, because me as the first speaker, and then Rob can directly jump into the real science. And I think Rob, he had also a slide on where serotonergic neurons are located. I have a slightly different one here. It's here, so we know in the mammalian brain we have serotonergic neurons in the so-called DRN, dorsal raphe nucleus. And perhaps not so easy to see here, but they project all over the place. These arrows here are thought to be axons, projecting into many different brain areas. And if you now zoom into one target region here, somewhere, let's say in the prefrontal cortex, but doesn't matter, it's kind of the general principle, you can look at this synapses. I am not sure about the background or all of you in terms of sciences, neuroscience, so I thought I give a very broad initial introduction. So you have this axon. At the end of the axon, you have the terminal here with the transmitter, the synaptic cleft, which was a discussion for quite some time in neurosciences, for about 100 years, whether everything is directly connected or whether there are these gaps, units. And then you have these kind of proteins here, which are the receptors. And you can modulate the serotonergic system. And the classical way people do in the clinics right now is blocking these uptake of via the transporter here. And there you can slowly and slightly increase the serotonergic level, the serotonin level. You can do also a much stronger treatment, fast and strong. And I think Rob will talk about this. Mainly it's about MDMA. And you get this massive increase here in serotonin. And last but not least, what you also can do is you can bypass this and directly activate these receptors here. This is shown here, for example, for a classical, one of the serotonin receptors, 5-HT2A. And then you can also have, of course, dependent also on the concentration, increase the serotonergic or enhance the serotonergic system. And when I started, the neurons were localized and the first cellular effects have been described on neurons of serotonin. Actually, my previous supervisor was early on in the 80s working on this. But the receptors were not really cloned. And then all these receptors, were cloned one after the other. And I have to say, I was a bit nervous. I mean, most transmitter systems, they have then 1, 2, 3 receptors, but here are many. And they have even subclasses, 5-HT1A, 1B, 2A, 2B, 2C, and so many of those. I never gave up completely, and you will see I brought a small story with me today. We are still working and busy with, but it's a rather complex system, and I think the good news is we all have to do a lot of work. And this is now the story. So what we thought is, so how is serotonin affecting the brain? The synapse, this is something we worked on a lot. It can modulate the synaptic transmission. It can directly affect single neurons. And it can affect, and the question was also for us, will it really affect also the overall network of the brain? And what we did is doesn't matter, the resolution for whatever reason is a bit down here. But what we did is we started to record in a brain area, which I would like to briefly introduce to you, which is the so-called medial entorhinal cortex. This was actually the second part of my PhD. At that time, it was very little investigated. So people thought, oh yeah, that's simply an input into the so-called hippocampus, which is the most, I think, investigated brain area, perhaps. And we started to characterize different cell types, and not because of me, because of others. It's a quite well-known area. Actually, 2 Nobel Prizes were given now for the discovery of so-called grid cells, which are quite specific for the entorhinal cortex or the neighbor structures. It's located here, very deep in the brain. It's not so easy sometimes to target. And what was also for us interested, it receives lots of serotonergic inputs from the nucleus I was talking about into the, it's a heavy input into the entorhinal cortex, and there are also tons of receptors. And what I also found exciting is it's a kind of, some people would call it the multimodal center. We receive tons of this inputs and outputs into this entorhinal cortex area. And what it does under baseline conditions in head-fixed anesthetized animals, you will find a kind of a brain rhythm. Some people call it the default mode, a kind of an intrinsic mode. We're working a lot on oscillations, and it's still not clear how this oscillation is coming about. But what we wanted to do is to study how serotonin affects this baseline brain rhythm. And we use different methods, and I will guide you through. So we are physiologists, so we do the so-called patch clamp technology, single cell. We try to do in vivo recording, pharmacology, genetics, optogenetics, and we use genetically modified animals. And this is the first recording session we did is with an electrode, which is called silicon probes. This is shown here, and we introduced several of those into the brain, here into the medial entorhinal cortex. To be honest, initially we sometimes failed and didn't get it in the right position. And then you can record for many neurons at the same time. And these are these action potentials, the spikes of the cells. And what we did then, we applied after kind of a baseline situation, this is shown here for the up and down states, and for the spikes, then MDMA. And I have to say, I mean, sometimes we work on these kind of small effects, 10, 20, 30%, but this was, okay. But this was really massive, I have to say. I've rarely seen such an effect. It's completely wiped out. It's gone. These up and down states, so this is true here for the up and down states, and also these spikes we were measuring. And then after washout of the MDMA, it's slowly recovered. So the animal actually and the cells are still alive. It's good to have a recovery. And here we plot a kind of the time course for this effect for MDMA. So you have a baseline, which is usually without MDMA, quite stable. This we also, it's a kind of an intrinsic control because we were using anesthetics to be sure that there is not a kind of a spontaneous rundown. And as you can see here, MDMA, as shown before, it has this massive effect both on the up states and also on the spikes. And then we did an additional experiment because MDMA is not only releasing serotonin, it will also release oxytocin, it will also release dopamine to a lesser extent, but who knows, yeah? And we used another substance which is called fenfluramine, FFA. And as you can see here, this FFA, here in kind of yellow, orange, it's more or less mimicking this effect. It's also completely wiping out here this activity in the entorhinal cortex. So we are mechanistic people, so we thought, well, this is a nice starting point. So now we have a big result here, a strong result, strong effect. Let's look into the underlying cellular or network mechanisms. And these in vivo situations are, on one hand, it's a kind of the real world, so to say, but on the other hand, it's technically sometimes a bit challenging. So one method we used in the initial part of the study was, it's called spike sorting. I don't want to go too deep into it. It's also not perfect, but you can get roughly in terms of the spike shape here, whether it's broader spikes, narrower spike, this so-called repolarization, you can get an idea what kind of cells might be involved. And we saw that these cells which broader spikes, putative, excitatory cells, principal cells, projection neurons, you might call them, were mainly not affected by MDMA. Then there were cells which have a very narrow spike. They are so-called GABAergic interneurons or subclasses of GABAergic interneurons. Some, they are named basket cells or path basket cells. They have very fast spikes, 5 times faster than the others. But there was kind of an intermediate group. They were not broad, they were not very narrow, and those were the cells which massively increased with MDMA. So we thought, okay, what's going on there? What could this be? Again, this quality is not great, but I think I can guide you through here. So what we did then next is to get a more handle on what might be the underlying mechanism. We switched into another method, in vitro electrophysiology. What you can do is you can cut brain slices, and then it's easier to do cellular recordings, to do identified recordings, to do pharmacology, and so on. And first, and thanks to Prateep, we had another study going on, which was the requirement actually to do this. We had to establish these kind of brain rhythms, or to be able to measure these brain rhythms in vitro. And what we did, we recorded with the patch clamp technology from several cells at the same time. Here we have setups where we can do up to 8 cells at the same time. Here's an example with four such cells. And you see there's activity in all these 4 cells, spikes, subthreshold activity, and this is highly synchronized if you do this kind of analysis. And if you now in vitro apply MDMA or serotonin, so a kind of a mimicry, so the ligand, which the 5-hydroxytryptamine, which we did here at relatively low concentrations, and on all the slices and cells we investigated, you see this massive effect of serotonin. Here it's a bit delayed, but it's also going down, and here you see all these individual recordings, and yeah, here's a summary plot. Again, in vitro, I guess we can mimic the situation from in vivo with serotonin. It's kind of reassuring. You get this massive reduction. Then we thought, okay, which receptors might be involved, which is something I sometimes don't like to do because the pharmacology is a bit tricky. So many drugs are out there, and but we were, I would say, partly lucky. We started with 5-HT1A, there was no effect, 5-HT3, no effect, but 5-HT2A agonists, so also the receptor where usually psilocybin or psilocybin or LSD would bind to, not exclusively, but mainly. And we used another drug here, it's called alpha-methyl 5-HT. It's a relative selective 5-HT2A agonist. And you can see we have less ends here, recordings, but it mimics the effect of serotonin. So it looks like that at least 5-HT2A is involved. And we had also done some antagonist experiments with ketanserin. And to be honest with you, it was reduced. The effect was clearly reduced. I have also one slide on this, but it was not completely gone. I mean, it sometimes might be other receptors, might be also that it's a matter of concentration. And now coming back to the the cell types being involved into this effect, and I will cut this a bit short. So what we initially did, because the entorhinal cortex is not fully characterized as other brain regions, there are tons of different cells which we still don't understand fully. We had a kind of a massive brute force recording for many cells, and again, in vitro, we had the result that these kind of medium board spikes, these cells are sensitive to serotonin. And then what we did is we used a kind of a transgenic approach. We had animals, many different types of animals, path interneurons, but also so-called somatostatin. It's a subtype for interneurons, quiline, and we brought in first a reporter. It's called here TD tomato, so you can identify these cells here in the entorhinal cortex. And then what you can do is you can do this kind of targeted patching. So here's a slice, looks like a slice of bread here actually with a biorender, and you patch these cells here from the transgenic animal. And if you do so, we can characterize the cells. So it looks like GABAergic interneurons. And as you can see here with serotonin, you get a clear depolarization of the cells, which is not the case for the principal cells. There you have a slight hyperpolarization. So a clear, strong effect. And this effect is antagonized by ketanserin, the 5-HT2A receptor antagonist. So then next, what we wanted to make sure is, I mean, one could call it control experiment, but I think it's a bit more than a control experiment. We used an optogenetic approach to make really sure that the somatostatin interneurons, which initially was a bit hard for us to believe that they are really so effective and so strong there, because the focus in research was so heavy on other interneurons, we used again a kind of a genetic approach, an optogenetic approach. So we used these mice and we brought a light-sensitive channel into these cells. This is something a great biophysicist here from Berlin discovered, and somebody from Stanford. They both did great work on this technology. It's very helpful in system neurosciences. So we brought this into these cells, and now I have to guide you through this slide. So we record here from cells, sometimes several cells at the same time, and then you get these kind of up and down states. And now you switch on the light, and whoop, it's more or less gone, the activity. And then turning off the light, the activity comes back. And this you can also then nicely show here in this raster plot for spikes and up and down states. Then I think I run out of time, so second last slide. We also thought about the other way around. So we used a kind of an antagonistic effect, not pharmacologically, but so-called chemogenetically. This is more for the experts perhaps here. We did a kind of a crossbreeding with two lines. Actually, this was homemade and actually also done in our lab. Crossbreeding with with the interneuron here. And what you can do then is we first had the effects of serotonin, and then you can activate these receptors. I mean, it's very specific, but what you can see here is that you can bring the activities then back. So the idea being that first serotonin would affect, perhaps depolarize these interneurons, and now you take this back, not to baseline, didn't come back completely, but it was a clear effect, I would say. We did also the other way around, first the chemogenetics, then serotonin. This, I would say, was quite clear. All right, I think I have to come to an end here, but I want to thank the people. So Prateep, I mentioned, he was the one who's setting up really the up and down states recordings in vitro. He was also the one training Roberto, a PhD student in our lab, and he did the MDMA study. And let's also finish with another anecdote. My last 2 minutes. I started with kids, and I also will conclude and finish. Here, these drawings here from my kids, Hannah and Lena, and they were 5 and 7 years old. And these are the so-called hippocampus. So these seahorses, which is the brain area we are mainly working in. And they did these drawings, which often on seminar, lecture series, we use this as a background, which is nice. But the story goes like this. So I was at home and putting some slides together, and was some computational work. I was on my way to Jerusalem, Hebrew University, and they have very strong mathematicians there. And I was a bit nervous, I have to say. They can give you really a tough time. And said, what are you working on? She was 5 years old. And I said, oh yeah, this is the brain, a bit of disease. Actually, there was nothing on disease, actually. But, and okay, talk was mediocre. And few months later, again, I was doing a few things at home, and I was on my way to Zurich. And again, Hannah was there. It was a Sunday. Oh, what are you doing? And to be honest, there was not much modification. One data slide left and right, and one additional perhaps. And what are you working on? I said, well, it's about the brain and disease. And she looked at me, mouth open, said, what? You are still busy with the brain? And I mean, for me, it was a kind of an eye-opener. She was, of course, very frustrated with her dad. She thought, oh my God, this guy is so slow and dumb. Why is he not working in astrophysics? And but for me, it was clear. So we do little things, at least we do little things, but as a kind of a collaborative community, I think we can do more, and some people are a bit faster than I am. Okay, so thanks so much for your attention.
Yeah, we can take 1 or 2 questions.
Okay.
So questions right now. We can also have the questions later if there's no burning questions immediately. Any questions? 1 question?
What do you think? What do you think that made your result? In astrophysics.
Yeah. I mean, I mean, for us, as I said in the beginning, for us is the investigation of serotonin on neurons. How is serotonin? It's a kind of a really basic research. How would they affect neurons, the synapses circuit, and so on. I can't tell you whether it has anything to do with behavior. Well, this is something we would still love to do. And I also can't tell you that whether psychedelics or MDMA would cause some of their effects via the entorhinal cortex. So still work in progress, so I can't say. But it's, I would say, a starting point. We have a big effect.
Thanks, Dietmar, very much. So now we move on to the next speaker, Professor Robert Malenka. He is here all the way from Stanford, and he will talk about mechanisms of the prosocial actions of MDMA. Thanks, Rob, for coming.
Thank you. Okay, well, of course, thank you, Prateep, for inviting me and organizing this session. And I can't tell you how pleasing it is as a basic neuroscientist to see how packed this this auditorium is, that there are people who actually care about basic mechanisms and what they might tell us about how this class of substances work. So I started my lab in 1990, before some of you were born, and I've always been interested in using drugs as powerful probes of nervous system function. For a while, I was studying drugs of abuse, such as cocaine and methamphetamine, as again, a powerful way of causing an experience in an experimental animal, and then delving into the brain and seeing what changes occur that account for the behavioral effects. But at least in the early 90s, I didn't think it would be prudent for my career to admit that I had an interest in studying both the classic hallucinogens such as psilocybin or LSD, and never mind a drug such as MDMA, which was getting a lot of bad press, especially in the Bay Area for a variety of reasons. But finally, I came out of the closet about 7 or 8 years ago and published a commentary with my then postdoc, Boris Heifetz, who's now an assistant professor at Stanford and a close collaborator. And we pointed out that studying the actions of MDMA is not, really could provide insight, not only to how MDMA works, but because of its very powerful pro-social effects in human beings, might actually tell us something fundamental about, I think, some of the most important and powerful components of human interaction. And towards the end of my talk, if there's time, I'm going to talk about empathy in mice and how MDMA might actually influence that. Of course, as a psychiatrist who once saw patients, I was also very interested in elucidating the actions of MDMA because, as you all know, it's being tested in a variety of clinical trials, most notably the MAPS trials and using MDMA as an adjunct for psychotherapy. Okay, so Boris was a postdoc in my lab. We teamed up, and he started doing mouse behavioral experiments. And the first thing he had to do was develop a simple behavioral assay for what I'm going to call either social preference or sociability. And the assay he chose to do is actually a fairly standard one. Sorry, I'm having trouble with. Oops, sorry. I can do this. Where is the button? Help me out. No, that's the slides. Did you get it? Which one? The left one? This one. Aha, I did it. Whoops, no. Sorry. Which one? Up here. I'm not going to use the goddamn pointer. All right. So he developed a behavioral assay. He didn't develop it. He used one that's standard in the field, shown in the upper left, which is known as the 3-chamber social preference assay, where you put your mouse in an experimental apparatus. It has 3 rooms. In one room, there's a little cup with a juvenile mouse, and in the other room, there's either an empty cup or a cup with a toy mouse. And you basically ask your subject mouse, which room do you want to hang out in? With the assumption that if it hangs out more around the cup with its little buddy, that's showing social preference or an increase in sociability. So in the upper right in B, what Boris did is a dose response of the effects of MDMA. And what you can see in a dose response, dose-dependent way, MDMA enhanced social preference or sociability in this admittedly very primitive and simple behavioral assay. In C and D, what you'll see is just the time course of the effect. And then in panel E on the lower right, one of my favorite experiments. And by the way, those of you who heard this talk yesterday in Charité, I apologize. It's the same talk. I was too lazy to put together a new talk. But maybe you'll learn something the second time around. So if you look at the bottom right, it's again, one of my favorite experiments. If you look below the graph, you'll see in the second bar, if we gave the MDMA to the mouse that was under the cup, there was a trend towards increasing sociability or social preference. If we gave the MDMA to the mouse that was free to explore, which is the standard way we did this assay, we saw a pretty clear increase in sociability or social preference. And then the last experiment I call the rave experiment, if we gave both mice MDMA, we had the biggest effect, which is actually, it's cute, but it actually may be telling us something about how MDMA actually is working. Okay. So one of the goals of this original project that was published now several years ago was to see if we could mechanistically, either at a molecular level or at a circuit level, dissociate what I'm going to call the pro-social effects from MDMA from what I'm going to define operationally as the abuse liability of MDMA. And what do I mean by that? MDMA is a psychostimulant. It's a part of the same family as methamphetamine and cocaine. I don't think it has nearly the abuse or addictive liability of those drugs, but nevertheless, because it's a psychostimulant, I think it has that potential. So, and I think that's a particularly important point, and Rick Doblin doesn't like it when I talk about this, but the way MDMA is being used in the MAPS trials, I think it is very safe. It's used as an adjunct for psychotherapy. It's only given twice. but I also anticipate, should the FDA in the United States approve the use of MDMA, even with the black box warning on it, that there's going to start being a lot of off-label use of it. And that poses the potential for inappropriate use and perhaps some use that will do some damage. So in mouse work, it turns out that every drug really pretty much ever tested, not only the psychostimulants, but the opiates, nicotine, alcohol, all drugs that have some form of abuse liability have two behavioral effects. One is very simple. They just increase the locomotor activity of your subject mouse. So what's shown on the left is in a dose-dependent way, MDMA at 7.5 milligrams per kilogram did not cause an increase in locomotion, but a higher dose did. Perhaps more pertinent to, in quotes, abuse liability is assaying the rewarding property of a drug, in this case MDMA. And the gold standard for determining the reinforcing or rewarding quality of a drug is doing some form of self-administration under an instrumental conditioning protocol in your mouse. That's kind of a painful thing to do. So a poor man's version of looking at the rewarding quality of any experience, in this case a drug, is what's known as conditioned place preference, where as shown in the diagram, you give MDMA and you limit the mouse to one specific context, in this case, the gray room on the left. On another day, you give it a control substance like saline and you put the mouse in a different room. And then on the test day, you ask the mouse, where do you want to hang out? And you assume if it wants to hang out in the room where it had a drug experience, that indicates the drug is reinforcing or rewarding. And you can see at the lower dose MDMA was not reinforcing, but at the higher dose it was. Okay, so what are the molecular targets of MDMA? We already heard from Dietmar that it's well established that MDMA, its major molecular targets, and certainly not its only ones, but its highest affinity molecular target is the serotonin transporter, which we call SERT. It also interacts with the dopamine transporter, which I'll call DAT. It has been reported to cause the release of oxytocin via an indirect mechanism. And there's a lot of indirect evidence saying increases in oxytocin levels may contribute to some of the behavioral effects of MDMA. We're gonna focus initially on the role of the serotonin transporter and whether that's important for mediating this pro-social effect of MDMA. So the first thing Boris did is a simple behavioral pharmacology experiment. He gave MDMA IP, but before doing that, he gave a drug called citalopram, which is a high affinity binder to the serotonin transporter, which prevents MDMA from binding the serotonin transporter. And you can see that that prevented the pro-social effects of MDMA. He then took a molecular approach using the modern tools of neuroscience, meaning really a genetic approach. We obtained a conditional knockout of the serotonin transporter, which allowed Boris to delete the serotonin transporter only from the dorsal raphe neurons that produce serotonin. And you can see in panel E, that knocking out the serotonin transporter from dorsal raphe serotonin neurons, it's noisy data, but on average blocked the pro-social effects of MDMA, saying its molecular interaction with the serotonin transporter is necessary. Importantly, in those same mice, if we used a higher dose of MDMA and assayed its reinforcing quality in this conditioned place preference assay, that was not affected by knocking out the serotonin transporter. It also wasn't affected by citalopram. So right there, that tells you that the molecular target that is mediating the reinforcing quality of the MDMA experience, it does not require the serotonin transporter. Okay, so where is MDMA acting in the brain? I hope most of you are neuroscientists and know there's something called the classic mesolimbic reward circuitry, which classically consists of dopamine neurons in the ventral tegmental area that send projections to another key component of this reward circuitry called the nucleus accumbens, also known as the ventral striatum. I didn't have time to go through this, but my lab has done a lot of work doing manipulations that suggest that the release of serotonin, specifically in the nucleus accumbens, promotes prosocial behaviors, promotes social reward, promotes sociability. So given that MDMA was interacting with the serotonin transporter, and I'll show you later, causes a massive release of serotonin in the nucleus accumbens, it was an obvious experiment to test whether directly infusing MDMA into the nucleus accumbens, this is old-fashioned behavioral pharmacology, but I still think there's a place for that in modern neuroscience. And what you can see in the purple bar here is that direct infusion of MDMA into the nucleus accumbens had a very robust enhancement of sociability in this 3-chamber assay, saying that its action only in the ventral striatum is at least sufficient for this specific behavioral effect of MDMA. Doesn't mean for many of the other effects, nucleus accumbens is the target. He also did the converse experiment where he gave MDMA IP in the belly of the mouse, and he infused the serotonin transporter antagonist, or the inhibitor, citalopram, directly into the nucleus accumbens. So this is just a test of this hypothesis that MDMA is acting on SERT within the nucleus accumbens. And you can see in the right bar that on average, this manipulation blocked the prosocial effects of MDMA. So we have 2 different behavioral pharmacology experiments suggesting it's MDMA interacting with the serotonin transporter, most likely on the serotonergic inputs in the nucleus accumbens that's required for its prosocial interaction. Importantly, this is another condition-place preference assay, infusing citalopram into the nucleus accumbens to prevent MDMA from interacting with the serotonin transporter. It did not block the condition-place preference caused by MDMA, whereas a non-specific dopamine receptor antagonist infused in the nucleus accumbens did, which is not unexpected. This is just a panel from a paper we published in Neuron recently on a related but different topic, where we used a genetically encoded sensor of serotonin, which we expressed in neurons in the nucleus accumbens, and then we gave the animal MDMA IP into its belly. And all this what this shows you is that, as expected, MDMA does cause a massive release of serotonin in this target structure because it receives a pretty heavy serotonergic innervation. So, of course, we were interested in the potential therapeutic potential of MDMA. I have a specific interest in autism spectrum disorder for a variety of reasons. So we, in subsequent work done by Jessica Walsh, she obtained actually 6 different mouse models of autism spectrum disorder. I'm going to show you data from 3 of them. This is, these first 2 are genetically based models of autism. What's graphed on the upper graphs is a different assay. It's called the juvenile intruder assay. I'm going to go for 25 minutes and leave 5 minutes. I'm just seeing you test it. I know I always go over. I'm well known for going over. But I flew a long way from California. And I am jet lagged. All right, so the top graphs are, it's a very simple assay. It's called the juvenile interaction or intruder assay. You take your subject mouse, you place a little buddy mouse in its cage, you use a juvenile so you reduce the normal aggression between conspecifics of the same sex. And what you can see, if you look at the blue bars, you can see that both in that assay and then the bottom graphs show the assay we've been doing, the 3-chamber social preference assay, that these autism mouse models have a deficit that is largely rescued by giving MDMA. And then this is a 3rd mouse model of autism. This is an environmental model where you give pregnant moms valproic acid, and for reasons I don't understand, the pups show deficits in a variety of behaviors related to autism spectrum disorder. And again, what you can see in the blue bar, in the blue lines, is these mice show a deficit in these sociability assays that's rescued by MDMA. How is MDMA working? It's going to be complicated, as Dietmar beautifully showed you. Serotonin causes the massive release of serotonin. What did I say? MDMA causes the massive release of serotonin. I think there are, 16 or 17 different serotonin receptors in the brain. So it's really hard to figure out which of those receptors the released serotonin caused by MDMA is critical for the behavioral effect you're studying. For a variety of reasons based on previous work, we were specifically interested in a receptor called the serotonin 1B receptor. And what this shows you is that if we infuse a 1B receptor antagonist directly into the nucleus accumbens, we can block the behavioral effects of MDMA. Finally, and this is a plug for colleagues here from MindMed who are pursuing the possibility that different enantiomers of MDMA may have therapeutic benefit. So why is that? The MDMA that hopefully you guys don't buy off the street but get from a reliable source, and more importantly, the MDMA that people are using in clinical trials, in particular MAPS, is a racemic mixture of two different enantiomers. It's RS-MDMA. And if you go into the literature and you look at the affinities of the R versus the S enantiomer, you look at some of the behavioral assays that have been done, it looks like the R enantiomer has a higher affinity for the serotonin transporter and a lower affinity for the dopamine transporter. And based on our hypotheses, that would suggest that the R enantiomer should still have prosocial effects, but might have a reduced abuse liability. And this just shows in a small number of animals, it's only like 5 or, well, it's a very small number, that our MDMA did enhance sociability in this simple assay. So there are actually 2 clinical trials going on based in part on this kind of work. A company called MapLight is pursuing a serotonin 1B receptor agonist in autism, and MindMed is pursuing doing early work to try to get an IND around our MDMA. What does this have to do with empathy? So, as some of you know, MDMA, people in the field, I think, disagree and argue whether MDMA is an empathogen, meaning it actually enhances, in quotes, empathy, however you want to define it, or as you know, God, who coined the term? Was it Shulgin who called it an entactogen? Is that right, Matthias? Oh, it was David Nichols. I'm sorry, it was David Nichols. In part because does it actually influence empathy? Does it actually influence how I interact with the person in my environment? So we decided to see if we could develop behavioral assays of what I'm going to call behavioral antecedents of empathy in mice. And I'm going to tell you very quickly in the last 5 minutes about some assays, a postdoc in my lab who now has her own lab at UC San Diego, which is called the social transfer of pain. And it's actually a fairly simple assay. You take 2 mice, you put 1 mouse in pain by injecting a toxic substance in its hind paw, and then you take another mouse, which we call the bystander or observer mouse, and you just let it hang out with the mouse in pain for an hour. And then that observer mouse will exhibit a bunch of different behaviors that indicate that it is experiencing pain. And one way of showing that on the lower left is doing a mechanical sensitivity assay it's a very standard assay in the pain field using things called von Frey, whatever. Again, what you can see on the lower left is the animal with CFA has a very low mechanical threshold for withdrawing its paw, meaning it's in pain. The purple line shows the bystander mouse. So this is a mouse that has never experienced physical pain, just hung out with another mouse in pain. And you can see it manifests behaviors indicating it's actually in pain, and these other graphs just confirm that, for about 4 to 12 hours. We also developed an assay of sort of the opposite effect, and this is the social transfer of analgesia. So in this assay, both mice are in pain. So both mice have received an injection of a toxic substance in a hind paw. So they're limping around. We then give 1 mouse morphine. So it's analgesic. It's no longer experiencing pain. We take our other mouse who's in pain and just let it hang out with the mouse that is experiencing analgesia. And what you can see is that its pain threshold has increased. Its mechanical sensitivity has decreased. That's the blue lines. So it's pretty wild. But again, this should be intuitively obvious if any of you have kids, for example. If you're a parent and your kid is sick, it makes you feel awful. You want to take that pain away. So I am anthropomorphizing this a little bit, but I would argue that these are behavioral antecedents of what we term empathy. So does MDMA affect this? And then I'll wrap up. So this is the key result. This is hot and not published yet. So this is just the pain threshold, the mechanical threshold in the mouse that's in pain. It has a very low mechanical threshold. Here's the bystander mouse. Here now, we only let the bystander mouse hang out with the mouse in pain for 10 minutes, a much shorter interaction period. And in this, for some reason, they show a little bit of increase in their pain, a decrease in their mechanical threshold that lasts about 1 to 4 hours. If we give the bystander mouse MDMA, we dramatically enhance that social transfer of pain. So that mouse experiencing MDMA is now much more sensitive to the affective state of this mouse in pain. And obviously we did control experiments. MDMA itself does not affect pain thresholds. What is this? I'll skip that. What is this? So this just shows you in a different way that direct infusion of MDMA into the nucleus accumbens enhances the social transfer of pain. So in the middle bars, graphs on these slides where it says saline, there's a little bit of an effect. There's a little bit of a social transfer of pain, but not much. But in the animals that received MDMA infusion into the accumbens, there's a very clear social transfer of pain. And then, I know I'm going quickly, but the take-home message is we did the same experiment with the social transfer of analgesia, and MDMA IP enhanced the social transfer of analgesia. I don't have time to go through the bar graphs, but you can just see the blue bars are much higher than the other bars. And then finally, we again did the experiment of infusing MDMA directly into the nucleus accumbens, and again, that compared to infusion of saline in the accumbens, that enhanced the social transfer of analgesia. So I actually finished more or less on time, which is very unusual for me. So the take-home message is MDMA promotes prosocial behaviors and, in quotes, empathy. Empathy as operationally defined by these primitive social transfer assays in mice, at least in part by causing large increases in serotonin, specifically in this target region of classic reward circuitry. And this requires an interaction with the serotonin transporter. Serotonin 1B receptors in the accumbens are responsible for some of the effects of MDMA. It turns out the serotonin 1B receptor antagonist does not seem to be blocking the social transfer of pain, meaning there's some other serotonin receptors involved in that. And I would argue, and this is a good topic for the panel discussion, that the importance of this kind of preclinical foundational neuroscience work is maybe by understanding how a drug like MDMA works. Not only can we make better versions of MDMA with more of the therapeutic components of its effects and reduce its potential side effects, its potential toxicity. But I also think by using MDMA as probes, we may actually learn something about the neural mechanisms of empathy and perhaps even compassion. And I would argue what is more important in the world today, given the state of our world. So with that, I will end, and I'm happy to answer any questions. And the people who did the work, I just sit in front of my computer, so I don't do any of this. All right, well, 1, 2, sorry. Go ahead. Because it works. Operationally, it actually, we can quantify pain behavior using like these mechanical thresholds. It's funny, so you're asking, why didn't we look at the social transfer of reward? Is that, we're actually doing experiments it's not quite that. I have another postdoc, he's doing experiments. I don't know whether to call it helping behavior, compassion behavior. We're asking whether a mouse will learn to work to give another mouse a reward. So it's not quite answering your question, but and it looks like it's working. And then you can play lots of games with that. One other question.
So I noticed this might not have been statistically significant, but I noticed that there was one bar graph you showed in the beginning that showed that homozygous certain knockout animals under the influence of MDMA had reduced social. I wonder if you had any mechanistic insight into why that might be.
That's a great question. I go fast because I don't like people. It's a really, you pay attention to the data. You can work with me. Because a lot of people don't pay attention to the data. So what he's referring to is when we genetically deleted all the serotonin transporters in a homozygous knockout, it looked like MDMA was actually reducing. And the answer is, let's talk offline. I don't have a good answer for that. All right, thank you very much.
Thank you for your talk.